Hi guys, so this is going to be the review for topic 3, which is genetics. So with genetics, our main topic here is DNA. Just remember, I want you guys to remember that DNA is found in the nucleus, okay? So that's the organelle where you find DNA, and DNA is short for deoxyribonucleic acid. That is found in the nucleus. Okay, so there's a couple of jobs of DNA, a couple of things you need to remember. So DNA is important because it carries the instructions for making proteins, okay? That's its first job. It's because proteins are doing so many things in your body. So first job is that DNA makes proteins, and specifically by piecing together amino acids in the right order. And that order is dependent on the DNA's order. And the DNA is composed of 4 different bases. You will never have to write out the names of these bases. Just remember, if you see the bases, to recognize what letter represents it. So adenine is represented by the letter A, that's the first letter. Thymine is represented by the letter T, and guanine is represented by the letter G, and cytosine, the letter C. These guys are base paired, right? Because DNA is double-stranded, so each strand has a base on it. And the pairs are as follows. A goes with T, and G always goes with C. Just remember, all teachers go crazy. Like you guys drive me crazy from time to time. So just remember the base pairing rules. You see, if you can see it here, G with C always, T with A always. Just know that before DNA copies itself, before DNA divides, yes, it must copy itself, because in mitosis we know that we want to have genetically identical cells. With mitosis, the only way we're going to have genetically identical cells is if the DNA copies itself before dividing. For example, you have a cell that may have 12 chromosomes. If it divides without replicating, the 2 new cells will only have 6 chromosomes. Compared to the parent cell, which has 12, these daughter cells will not be genetically identical. So for these daughter cells to be genetically identical, the original cell will have to replicate the DNA, going from 12 to 24, and then the division has to occur. So when those 2 daughter cells are formed, each will have 12 chromosomes like the original. Then they will be genetically identical to the original. Meiosis doesn't make genetically identical cells. Remember, meiosis is the results in sex cells, the sperm or the egg. So remember, mitosis makes body cells. Meiosis makes sex cells, sperm or egg. Okay? So if we're doing protein synthesis, this area here is describing protein synthesis, and I do hope you guys are jotting down notes. There's some additional things here that I'm saying that are not on this packet. But with protein synthesis, we know that the bases are read in groups of 3. Each 3 bases, so here you might have CAT, that might correspond to an amino acid, right? So these instructions are read in groups of 3, and each 3 codes, each 3 bases corresponds to one of 20 different amino acids. So according to the DNA, there is a certain protein that's being made. Remember, genes code for specific proteins, and the sequence has to be just right for it to make the right protein. And something to remember that's always helpful to remember, most proteins, actually all proteins, start with methionine. Methionine, or MET for short, is the start codon. Something to remember, okay? I guess you could remember the actual code, but I doubt that you'll have to remember the actual code. Methionine, just remember MET is the start codon. All proteins start with methionine, okay? Another important thing, okay, DNA always stays in the nucleus. When we're doing protein synthesis, DNA stays in the in the nucleus. DNA stays in the nucleus. However, when the protein is about to be assembled, an mRNA molecule is made from the DNA, and that RNA molecule, right, that acts as like a recipe for the protein, it'll leave the nucleus and travel over to the ribosome, where the protein synthesis is actually occurring. So remember, the DNA stays in the nucleus, but it helps make an RNA molecule called an M-messenger RNA, which travels to the ribosome to direct protein synthesis, right? So when protein synthesis is occurring at the ribosome, this organelle, Another RNA molecule works with another RNA molecule, tRNA and mRNA, and they piece together the correct amino acids according to what is said in the DNA and thus the mRNA to make a polypeptide or an amino acid chain, right? And once it's folded, it becomes a functional protein, okay? Remember, DNA stays in nucleus. RNA travels out of nucleus to the ribosome and helps make that protein, okay? Remember, right, you have your chromosomes here. This is an unduplicated chromosome. This is a duplicated chromosome. Each of these sections represents a gene, and each gene codes for a protein, okay? Proteins are very important in doing lots of things in our bodies. So here's the thing. Genes can be expressed or not expressed. So when we're talking about early cells in the embryo, these cells eventually start to differentiate and become the cells that are found in your body, right? They become nerve cells, bone cells, muscle cells. The thing is, all of your cells have all of the DNA to become whatever they want to be. But when differentiation occurs, if a cell is going off to become a skin cell, it still has muscle cell DNA and bone cell DNA, but muscle and bone cell DNA will be switched off. They'll be not expressed. The only DNA, the only genes that are expressed in that skin cell are skin cell genes, okay? Those are the genes that you see. These are the genes where you see those traits, right? So a skin cell, like all the other body cells, has all the DNA, but the only genes that are expressed are those skin cell genes. All the other genes for muscle cell, bone cell, nerve cell, switched off, not used, okay? Remember, all your body cells have all of the DNA available. They can be whatever cell they want to be, but the only gene that's switched on is the gene that it has differentiated into. For example, skin cell, okay? Another example is that hormones, they can turn reproductive cells on or off depending on where a human is in their development, okay? So hormones, remember, they are chemical signals. Most of them are protein. A lot of them are protein hormones, okay? Mutation, any kind of error in the DNA sequence, right? So the DNA has a certain order to the bases, and if there is a change in that order or sequence, it is called a mutation, okay? Either an added base or a base being removed. This causes a mistake in the order of amino acid. Remember, because the DNA ultimately determines the sequence of amino acids, okay? And then when the amino acids are adjusted or changed, your protein is not going to have the right shape. And if the protein doesn't have the right shape, it will not work properly. Mutations can be caused by chemicals or radiation or UV light from the sun or even x-rays when you go to the doctor from a broken bone. That is why your x-ray tech will wear so much protective gear, because they're going to be dealing with x-rays all the time. They do not want mutations, okay? Remember that mutations can only be passed on to offspring if they are found in sex cells. If you have a mutation in your muscle cell or your bone cell or your skin cell, you cannot pass that on to your future children. That mutation must be found in sperm or egg, okay? Yeah, pretty simple. Sometimes mutations can cause cancer, okay? Not all mutations cause cancer. Some mutations can cause dimples, but some mutations can be harmful and cause cancer. And remember, cancer is when your cells divide uncontrollably. It's a problem because the cells that are dividing uncontrollably are cells that don't work properly. This can make you very sick. Mutations also cause genetic variation. So in this case, mutations can actually be useful because remember, genetic variation is very important and very helpful for the survival of a species. All right, if a species has genetic variation, if there's ever a disaster that occurs, it ensures that some individuals in that species will survive, okay? Mutations can result in good or bad changes, but genetic variation is always good. You don't want to have a species that is genetically identical because any kind of event can wipe out an entire species. And when an entire species is wiped out, the ecosystem suffers. You reduce biodiversity and you reduce ecosystem stability. Guys, especially if I start talking slow, it's time to pause and write it down, right? So you're supposed to actually be scribbling down some notes on here. I know it's boring, but we got a Regents to pass. guys don't have to deal with me again next year. Selective breeding. This is why we have all these variations of dogs. Humans long ago, they found traits in certain dogs that they liked, and they made those dogs have offspring to give rise to more of this trait. For example, I'm not too familiar with dogs, like curly ears, right? If you have two dogs with curly ears, you want more curly-eared dogs, you're going to make those two dogs, hopefully boy and girl, make babies, okay? And those puppies will have curly ears, right? Whatever the favorable trait is, if you have two individuals of a different gender to have the mate, you can actually make this trait more prevalent. And then you see here, recombinant DNA. You see the word combine in recombinant DNA? If you ever have a question about it, if you ever forget what this means, you see that word combine there. That is just two DNA from different species being combined. So this can be made by using restriction enzymes to cut the DNA at a certain sequence. Okay, restriction enzymes cut DNA at certain sequence. Remember, enzymes are proteins. If the Regents asks you, what are enzymes made up of? Please say amino acids, okay? So, guys, make sure that you remember that recombinant DNA means combined DNA. So the main example here is human insulin, very important. So we use bacteria most of the time with recombinant DNA because bacteria reproduces very quickly. So whatever you're trying to make is going to be made very quickly if you use bacteria. So using a restriction enzyme, the gene for human insulin is cut out and introduced into a bacterial plasmid. That's another way to say bacterial DNA. Bacterial DNA is around. So you introduce that into the bacterial DNA and you put that back into the bacterial cell, and that bacteria is going to, through mitosis, because remember bacteria make clones of themselves, that bacteria through mitosis is going to multiply and multiply and multiply. And each of these cells is making the protein insulin. So this is how we mass produce insulin, okay? And I think currently this is the process still being used. We're still mass producing insulin this way. So we use bacteria because bacteria reproduces quickly via mitosis. Bacteria clones itself very quickly. What else? Gel electrophoresis. All right, I feel like this is used a lot in crimes. Usually the DNA of suspects are compared to the DNA of whoever committed the crime, whoever's DNA was left at the crime. But this is how it works. Fragments of DNA are separated by size. Right, and the DNA fragments are moved along a gel using electricity. Right, so restriction enzymes cut the DNA into smaller segments, and they are cut in various sizes. The DNA is loaded into a gel, and the gel floats in a buffer sequence between two electrodes. So on the negative side, that's the top. The positive side is the bottom. So when an electric current is passed through the chamber, the DNA will move from negative to positive. Now the bigger sequences will move slower, and they'll be closer to the negative side. And the smaller sequences, or the smaller segments, will move faster, and they'll be closer to the positive side. So what's going to happen is these guys, each piece of DNA is going to create a pattern. And the DNA samples that are most closely related to each other will create a similar pattern. And then you can compare their relatedness, right? So if one of the suspects is the person who left the DNA, then you'll see that the banding patterns between the DNA left at the crime scene and the DNA of the correct perp, you'll see that those banding patterns are the same. It'll be the same DNA, telling you that it's the same DNA. Okay, so that's gel electrophoresis. Just remember, DNA is separated by size, and that's it. Let me know if you have any questions. You should be working on your Regents questions right now, and I hope you took notes, because there's a couple of things that I said that were not on here. Okay, have a great day.